Okay, well, I hope you had a good break week, and I hope you have got some of their midterms out of the way. A couple of them, there's one more to go. Um, the topic of deficiencies, I know that we talk about it in plant science too, but I go over it again because a lot of students seem to forget what the symptoms are, and these things are important for you to remember, at least the major ones. So I go over it again, a little more detail with some more examples, and then I'm talking a little bit about toxicities at the end of the this talk. So let's get started. Terminology that you need to know and look for with deficiencies. Again, some of this should be review. Things like a purple petiole, right? So the petiole of the leaf showing purple pigmentation. Um, Intervenal chlorosis or chlorotic veins, necrotic spots, which is a tricky one because I think in your plant pathology you've come across leaves that have necrotic spots, and that is often more associated with toxicities or fungal infection or insect damage. But let's keep it on the list. Then there's chlorosis of the margin of the leaf on the outer edge, or tip burn, right? So things to remember. Again, I think I mentioned this before that macronutrients tend to be mobile and micronutrients tend to be immobile. So your NPK, it's no surprise you find them on this side. We rarely talk about chlorine and nickel, but we do talk about magnesium and sulfur. And these are more along, especially sulfur, on the line of somewhat mobile. So we'll keep that in mind as we talk again about where to find it. And remember, the reason why we talk about mobile and immobile, is because if a nutrient is mobile, that means that it can be reallocated. It's mobile. It's like, if you don't like your apartment, you're going to pack up and move to another apartment. So mobile nutrients, if the plant is not happy with where you are, it will take you and move you to another location. And usually when there's a deficiency, i.e. not enough of a particular nutrient, the plant will look at other tissues that have that nutrient, and if it's mobile, they'll remove it and then relocate it to where it's needed, more importantly, like the apical meristem. And so things like nitrogen, phosphorus, potassium, these are mobile, and if the plant's running short in the roots or not bringing enough in, then they will extract it from old leaves at the bottom that don't get a lot of sunlight or maybe less efficient. We'll remove them and then move them up to the top where new leaves are forming. So it's sort of a self-preservation move, right? So that's why we talk about mobile and immobile. Some nutrients, like all these ones, are built into the plant in such a way that the plant cannot extract them anymore. So once they've been used, it's done. They're stuck. It's permanent. So these are the ones that are especially troublesome for plants. And fortunately, other than maybe iron and calcium, they're not needed in much supply. We talked a little bit about boron deficiencies with poinsettias because they seem to be inefficient at taking up boron. But for the most part, it is calcium and iron that you will see in your lives. So let's review some of the obvious ones from plant science too. Nitrogen, which is mobile, right, causes general yellowing of the lower or older leaves. So your classic house plant is got yellow lower leaves and dropping leaves all the time. The ficus at the mall is dropping leaves because they don't feed it, they just water it. So when you don't have enough nitrogen, plants will disassemble chlorophyll, which has lots of nitrogen in it, which makes the leaves go yellow, and then they'll move the nitrogen up to the top of the plant where the new leaves are forming. So nitrogen, mobile. Nitrogen deficiencies, obviously, they present at the bottom with yellowing. Even the top leaves will be less green because there is less nitrogen to make chlorophyll. But generally speaking, the bottom is yellow, top is light green. Really good example here. This is actually like a canola plant. This is an orange tree showing yellowing on older leaves. When it's a mature tree, it's harder to tell old from new, but You sort of look at each branch individually. Don't look at the whole tree. Um, Phosphorus. You should also remember this. I think in labs, when I've asked you guys, you've for the most part remembered. The key with phosphorus is purpling, right? And it is older leaves, again, because it is mobile. So it starts in the petioles often, but sometimes it's in the leaves. In the case of tomatoes, we see it in the leaves sooner than we do in the petiole, as you can see in these photographs. The phosphorus is fairly straightforward. And when you don't have enough phosphorus, one of the key things is the plant is stunted. Internodes are shorter. And we talked about that with the crops class about growing plugs, right? There's some phosphorus deficiency outside. One of the key things to remind you of is that phosphorus deficiencies often present in cold temperatures. So when you're growing outside, like in this photograph, this could be just because there was a couple of cold nights, and plants are very inefficient at extracting phosphorus from the soil when it's cold, so they go purple. Strawberries, which is one you might see in the greenhouse, they are tricky with phosphorus, and you get this sort of purpling on older leaves. That's one to keep an eye on. Okay, let's move on to potassium. Again, this is review. You're looking at, it's obviously mobile, interveinal chlorosis in the older leaves because it is mobile. It often starts in the margins, like on this tomato leaf here, and then progresses to the entire leaf. And it all becomes necrotic. In cucumbers, you see cucumbers are a little tricky because there's a couple of nutrients that cause this sort of yellow haloing of the leaf. You see this bright yellow that's forming. But generally, we like to say that that's a potassium problem. It can also be related to chloride, and that's for the most part chloride. But let's just call it potassium for the most part when we see that. And pepper plants, they have leather leaves, sort of thicker leathery leaves. They tend to show up as marginal necrosis on the edges, as opposed to in interveinal margin necrotic spots. So it's much more marginal in the peppers. This is a close-up of a tomato plant showing early onset of potassium deficiency. So on the margins, and we're getting some yellowing, and then it'll become interveinal necrotic spots as it matures. And there's another picture of cucumber leaf showing potassium deficiency with the haloing or yellow chlorosis of the margins. And this is in an ornamental crop. It's a kind of a dogwood, very pronounced chlorotic outer margins of the leaves. Classic potassium. I think I mentioned this, that potassium is primarily used as an osmotic nutrient, so expanding cells. And when cells can't expand properly, they split, and that's how you get that necrosis. So they just become weak and die. Calcium. So calcium we've talked about, again, Plant Science 2, but be a little more specific today about things like blossom end rot in tomatoes and edge burn in lettuce. Cupping in cucumbers, we've already seen that. And extensive necrotic spots in cucumbers in really pronounced calcium deficiencies. With the lettuce, I want to mention just briefly the margins of lettuce leaves often become necrotic when you don't have enough calcium, and that is primarily because calcium is moved, that's immobile, right? And it's moved through the plant by transpiration. We've talked about this a bunch of times with poinsettias, with brack edge burn. The same idea here with lettuce. If you don't have enough transpiration, calcium doesn't make it to the edges of the leaves, especially the new leaves, where the head tends to be more folded in on itself and more dense, and you get higher humidity there. Remember, one of the things that moves water through leaves is transpiration, and if we have high humidity, we don't get transpiration. If there's no transpiration, water's not moving through the tissues. It doesn't pull that calcium to the edge of the leaf. So Really important with lettuce that you have fans for air movement to make sure that those delicate leaves have enough transpiration happening. You can imagine a dense iceberg head of lettuce where the leaves are all folded in over each other, that you're going to have localized high humidity that's going to make it difficult to move calcium. So lettuce is particularly prone to it. In the case of the cucumbers, this was when we had insufficient nutrient supply because it was a combination of nutrients and water. That's a photo from our greenhouse that I took. Sad. And blossom end rot, I think I have another slide coming up, but when the ovary is first forming in the tomato, the very end, which is furthest from the petiole, is more likely to have insufficient calcium. And so then the cells don't form properly and they're weak. And then as the ovary starts to enlarge, as the fruit forms, it tears the end of the ovary open, which causes necrosis, and you get that rotten bottom of your tomato. There you go, blossom end rot. So this problem started way back at the beginning, shortly after the flower is pollinated, like at this stage. So if you don't have enough calcium, even in the early stages, you're going to end up with blossom end rot. This plant actually has a lot of issues. Look at this dark margin here and light central parts of the leaves. Some interesting stuff going on there. Cucumbers will also get blossom end rot. A lot of times people just talk about tomatoes, but it happens in cucumbers. If you have the end of a cucumber rots easily or is soft, then you did not have enough calcium when that fruit was forming. And again, this is another picture showing calcium deficiency when the leaves are trying to form. And with tomatoes, the meristematic tissues can't form properly and the leaves are necrotic, especially the very young, sensitive tissues. They will die with insufficient calcium because those cell walls just can't form, and the new tissues are growing so rapidly, and if their cells aren't forming properly, they tear and rip and dry up and die. So yeah, calcium is very important. And a lot of it is managing your environment in your greenhouse, right? Making sure that it's not humid, not too humid, that there's enough transpiration happening on the plants. Yeah, so things like not enough nutrient being supplied or your humidity is too high. Because remember, calcium uptake in a plant is passive in that the plant can only move the calcium through water movement in the plant, which means that the stomata have to be open and there has to be actual evaporation happening from the plant. If your greenhouse humidity is too high, you're not going to have evaporation and you don't have water movement and you don't get calcium delivered to those tissues. Magnesium, mobile. Intervenal chlorosis, that's pretty straightforward. This is classic, what you're looking for. With peppers, same thing. Tomatoes, same thing. Intervenal chlorosis, pretty straightforward. Sulfur, from Plant Science 2, I use this picture as well because it's pretty obvious that the eggplant on the right has overall yellowing and on the left does not. Now, sulfur is what we call moderately immobile, which means it presents all over the plant, and this is species dependent, so you have to sort of do the research on your particular plants. Most plants present at the bottom and in the middle for sure, and lastly at the apical meristem. This is a pot mum on the right that has a sulfur deficiency. A lot of times we would say this looks like nitrogen deficiency, but that's actually a sulfur deficiency. So that can happen. If you're using city water and you don't have enough Epsom salt, sulfur deficiency is quite a possibility. Often well water has enough and surface water has enough sulfur in it that it's less of an issue. If you're collecting rainwater, that can be an issue too. So yeah, it resembles nitrogen, right? That's terrible. It looks like a W. Iron deficiency, again, we've talked so much about this, but I'm going to drive it home because you need to know this. It's got to be right on the tip of your tongue the second you see it, right? We know it's immobile, so it presents at the top of the plant. We know that it's general chlorosis. It can also be intervenal chlorosis like here, or just general chlorotic tissues like in a cucumber apical meristem. Typically it is insufficient supply, but remember the first thing we check is pH. All right, I know you said, oh, Derek, we know that, and I'm so happy that you said that because it means you know it. One more shot of cucumbers with iron deficiency. And again, just a reminder, pH is a key issue here. Zinc, immobile. So it's at newer growth first. Now this one's less, it looks a bit unusual. I think it looks quite a bit different from iron in that the veins are much more visible. So that's how I distinguish zinc from iron. But the other reason you might want to distinguish zinc from iron is because zinc deficiencies are rare. Like the chances of you seeing one are slim. But there you have it. That's what they look like. The veins are much more pronounced. I know in greenhouses that collect rainwater, it's never an issue because, first of all, rain is slightly acidic because it passes through the air, and the air has carbon dioxide, which is very water soluble. That forms carbonic acid in the rain. So rain always has a slightly acidic pH. Not crazy, but slightly. And that moderate acid in the rain, as it washes over the greenhouse, runs down the gutters to go to the cistern. And all greenhouses have galvanized steel gutters because of strength. And galvanizing is made of zinc. And the acid actually etches some of the zinc off over as the water runs along. And that's more than enough that for what you need for plants to have sufficient zinc. So typically in greenhouses, you don't see zinc deficiencies. Boron, we do run into. Again, we talked about this with poinsettias because boron is an issue, so is molybdenum. Boron is immobile, right? It's essential for meristematic tissues. So any new forming fruit or apical meristems, if you don't have enough boron, they become deformed. And on this tomato, you'll see that there's kind of a corky scarring around the top of the fruit. That is from deformed tissues in the meristem, and as the fruit formed, it tore. Now, unlike calcium with brackage bottom blossom end rot, which is at the bottom, the boron presents at the top of the fruit. So that helps you distinguish between boron and calcium when you're looking at tomatoes. One of the key things about boron is that it is passive. It is passively brought to the plant, just like calcium, which means it requires transpiration. So if you have a deficiency, you have to look at your climate to ensure your climate is correct, that it's active, that there's enough transpiration happening, the plants are actually evaporating water, that it's not too humid. So oftentimes cooler, humid times of the year, like the fall, can be an issue, or late in the fall, early winter when the greenhouse closes and your plants are still active, you get a lot of humidity buildup and you have to open the vents and vent that off or you end up with problems. This is another nutrient deficiency key. I'm not going to go over it. It's just another one for you to look at if you need a study aid or whatever. Let's do a little name that deficiency. So this is lettuce. What are we looking at? You got it. Calcium. So insufficient or insufficient air movement, right? That's the key with calcium. What are we looking at here? So I've got actually the next few are kind of tricky. What do we got here? So you see purpling and you see intravenous chlorosis. You see a lot. Yeah, there's a lot going on. So we know there's a magnesium deficiency because the intravenous chlorosis. Now this is assuming it's an older leaf. If this was an exam, I'd have to make that much more clear. But we also see purpling, which means phosphorus, right? We're getting anthocyanin buildup. So there's multiple things happening. When you see multiple deficiencies at once, the first thing you want to do is go to your fertilizer tank. Make sure that the A and B are being pumped out in equal ratios and make sure they're both stocked. Because often when you see sort of compounding deficiencies, there's some sort of fundamental problem that's much bigger than a minor deficiency. There's a big issue going on. Look at this little cucumber. What's going on with that? Well, there's a couple of things. Yellowing with fruit like cucumbers and zucchinis and some gourds, it's important to understand that a lot of them, their skin has chlorophyll in it, that the outer epidermis. And if they don't see enough sunlight, the epidermis becomes etiolated. In other words, the chlorophyll disappears and they go yellow. So sometimes the cucumber is tucked in the ground or it's buried under something and it gets yellow where it doesn't see sunshine. The second thing we're looking at here is this sort of pinch central hourglass shape. That means that there was insufficient water delivered to that fruit at a point in time. And it's important to understand it only takes a couple of hours of interruption with irrigation for a cucumber to not form properly because they swell so quickly. And once they swell, the tissue's kind of set and you can't fix it. So that there's two things going on here, right? So the fruit was probably buried under something which made it yellow, and there was a disruption in water supply causing the fruit to deform. What are we looking at here? Well, we have yellowing of the apical meristem, which tells us that it's an iron deficiency, but we have deformed juvenile leaves. This is I believe, a spinach that is not formed properly. So we're looking at boron as well. So this is another, I'm kind of doing these tricky combination problems because you run into these things. So we got iron and boron deficiencies going on here. This is interesting. We have a cucumber that has hollowed out, or the central axis has split. Um, this is often a combination of irregular water supply. Maybe someone's thought of that. Insufficient calcium, right? So as the ovary is forming, the cell walls are not properly attached, and as the fruit swells, it tears the center apart. And the other one can be insufficient boron, making the apical meristem weak so that when the fruit enlarges, the tissues are not properly attached. And lastly is excess nitrogen. When you oversupply nitrogen, the fruit swells and grows rapidly. This is sort of one of the classic things with nitrogen and potassium where we feed things like blueberries and raspberries and they end up being really big and we can fill containers to the grocery store with far fewer berries because the berries are so engorged. And that's to do with irrigation and fertilizers, which unfortunately make the fruit less sweet because you take the same amount of sugar that's in a wild blueberry and then put that into a swollen blueberry that's been fed huge amounts of fertilizer and irrigation, and you get that sugar is diluted across a much larger berry, giving you the sense that it's not as sweet. When in fact, the same amount of sugar is in each individual berry. It's just there's a lot more water and tissue. So overfeeding fertilizer to fruits is something that California and Florida farmers are well known for. Let's look at nutrient toxicity now. So that is when you either oversupply a nutrient, so you put too much in, or you have over availability, as in pH. This is a common one with things like iron and manganese. Yes, pH is important. So just to remind you, this is a key thing here, right? When you're looking at nutrient problems, you want to make sure that it's over the entire crop. Because if you find one plant that has a deficiency, you have to ask yourself the question, what is going on to this one plant that it is experiencing this problem while the other ones are not? And if it is a nutrient deficiency, you don't find any insects, more than likely, if you look down at the roots, dig into the soil, cut the bag open, you'll see that there's a root infection, some sort of fungal infection or aphids in the roots that are parasitizing the roots and weakening them so they cannot take the nutrients up as effectively as the other plants in your crop. So you have to do some detective work when you're looking at problems like that. So whether it's a toxicity or whatever deficiency, this is something that you want to make sure logically. Is there something unique to this plant? Is the pH wrong on just this plant? Why? What could have gone wrong? The other thing, across different crops, it is important to understand that crops are not all the same. I mentioned this in one of my labs. Students that are interested in cannabis, it would be well advised to understand the history of the plant and It's obviously related to hemp, and hemp is actually used for remediating soils because hemp is very effective at taking up heavy metals and nutrients like that, which means that if you oversupply certain micronutrients, you can actually reach toxic levels in certain plants. And in fact, cannabis is a crop where things like iron and manganese, you have to be very careful that you don't oversupply because they will quickly show toxicity, which would be localized necrotic spots, purpley spots all over the leaves. That's a sign that you've got toxicity. Foliar feeding, which is something we've talked about, is another one that can be risky if it's not applied evenly, right? Or if it's not mixed sufficiently. Sometimes in the tank, they settle out. You go have coffee, you leave the tank, you come back, you start spraying again. It's settled, and for the next little while, you're giving a much larger dose than normal. So it's important to, whether you're applying it or mixing it, that is consistent. The other thing is your math could be off. So that's when you got to, there's no excuse. After this, you got to remember how to do that. Get the math right. Let's look at some toxicities. So manganese and boron toxicity, and this is sort of toxicities, to be honest, almost all look the same, but at least if you see this, you have a good idea of what you're looking at. So early stages are marked by specks on the leaves with reduced chlorophyll. Now, right off the top, if you're looking at this geranium leaf and you've been doing IPM with Graham, you might say, well, couldn't that be stippling from an insect that's feeding on the leaf? And you'd be right to say that. So obviously, the first thing you're gonna do is you're going to inspect the leaf, flip it over, look for insects. Are there mites under there? What's going on? If there's no sign of any insects, then you move on to, okay, is this nutritional or is it fungal? And then you have to get your magnifying glass out, look to see if it's fungal. Look at the conditions. If it's fungal, would it be the entire crop at once, or would it just be one plant? It would probably just be one plant at first if you're scouting regularly. But if it shows up on the whole row or section of your greenhouse, then you're more likely to say, okay, we have a nutrient toxicity here. So again, using logic, right? So these spots you see, these stippling spots, or even marginally necrotic spots, those are early signs of a toxicity. So as manganese and boron toxicity progresses, it looks a little more pronounced. You get much larger necrotic spots. It starts to look kind of like a potassium deficiency. And this picture doesn't show it very well, but you can also get yellowing of the apical meristem because if you have excess manganese and boron, they compete. They actually outcompete the root spots for iron to enter the plant. So by having excess, remember how we talked about antagonistic and synergisms with nutrients? Excess manganese and boron will actually be antagonistic to iron uptake. So you can also see a combination of necrotic spots and yellowing in the younger leaves as a result of excess manganese and boron availability, either one. And I'm going to say most likely manganese if you had to choose. Okay, I'm not going to promise, but I hope to be done talking about nutrient deficiencies. So if you're tired of hearing about it, maybe that's a good thing because it is an important thing to know about. So I've got some interesting links in this week's folder for you to look at. There's a paper, there's some interesting discussions, web articles. If you're curious, have a look at them. None of it's required, but might be of interest to you if you have time. I know you can be pretty busy now that you're back from break week and things ramp up again, but okay, see you in the greenhouse.